You guys see the screens okay? Awesome. So before we get started on the lecture component, I obviously wanted to touch on just the format of the labs. Also because the lockdown has been extended, which is probably not a surprise. And so we're just going to continue with the virtual format. Thank you for being patient as the lab portion of the faculty is trying to coordinate how to run those virtual labs. There was a lot of confusion with how we were going to do it and where they were going to post it. And it was confusing for me as well. So I tried to do my best to talk to them, get the most appropriate information, and then I made that announcement today just to sort of outline probably the best ways to do it. I don't think they ever told you that you guys needed to download LabChart, so that was probably super frustrating. And I got a lot of emails, and so I tried to do my due diligence and just try to break down everything for you guys. And so have you guys all seen the announcement on Canvas? Okay. So just read through it. It tells you kind of the locations of where the files are. You're going to watch those two videos, do the one quiz that's worth 1% of your grade. You're going to then do the report and then submit it on the turn it in option underneath. And then you'll be able to submit it. And then what was going to happen is that your lab reports are supposed to be due when you go in for the next lab. So you always get like 2 weeks. We can extend this one as to whenever you guys want to complete it, just based on the fact that there's been a lot of confusion as to even how to just download the files. I'm open to that. I just want to make sure that you guys sort of get the pattern of how to sort of coordinate everything to complete the report, because that would be similar for now the labs coming up. I'd imagine that we're going to be virtual for the rest of the semester. You guys want to bet with me? So any other questions in relation to sort of the lab component? So, sorry, I missed this. Just want to confirm the lab report is due on the 12th. It was supposed to be due like the next time you have the report. So let's say the labs were supposed to be your labs were scheduled for last week, the 21 and 22, correct? So then they would have been due the like 2 weeks from that point. They would have been due either the 5th or 6th, or sorry, the 4th or 5th. Or if you were scheduled to have your labs on the 28th or 29th, then they would have been the 11th or 12th. So you like whatever what your scheduled lab was, you were supposed to get 2 weeks from that point. Now, obviously, I can only post it all at the same time, so we can make it so that way you get 2 weeks. If you'd like. How do you guys think I should run it? Like, give a solid due date? Like, most of you are gonna say like no due date, which is I'm open to it. But then, how are you guys gonna know that you're going to? I gotta kind of give the TAs a little bit of a deadline, or else they're going to be frustrated because they want to get all their marking done for one thing, because they learn how to market, they do it, and then they switch to the next one. So I kind of have to give a bit of a deadline. So as a group, how would you guys what do you guys think? Obviously, this is confusion with this one, so I'm okay with pushing it back. What does Keisha say? Yes, grabbing the reaction time from the lab chart. Where exactly do we get that number? I give solid due date. Home is 12, maybe extended a week. Up to loss. I think solid due date. Okay, so Keisha, I gotta be honest with you. I have no idea, because they just did these labs now, right? Like I'm trying to coordinate it. So everyone, Keisha asks, when grabbing the reaction time from the lab chart, where exactly do they get that number? I'm assuming it's going to be, does anyone know? It's going to be a value.
Like he does it, like when he's showing us, it's not the exact time for each trial. So I just want to know like how accurate I have to be. Or could it just be a good guesstimate?
I feel like it could be a good guesstimate, but who's your TA? Because whoever your TAs is going to be the one marking it. So hopefully you guys can all hear this. I would email them and then they're not scheduled anymore, right? So they're open. So they would set up a Google Meets with you and then like show you. And that would be awesome if you did that, because then you can kind of report back to us. Not to put work on your plate.
Yeah, I'll do that then.
The TAs this semester are pretty nice. Like they were all my students before. Okay, wait. Scotty says in the video he just tries to eyeball the peak. I think it'll be different for everyone. Everyone's interpretation from the graphs will be slightly different. I think that's correct too, Selena. So Keisha, I would just confirm with the TA. And then in relations to the due date, I think we should make it the 12th. You get an extra week considering everything, and then I'll make an announcement. Lab is due the 12th. Then we'll have 2 weeks for the next lab, and so on and so on. How you guys all feel about that? Okay, cool. We good? Any other like sort of like bookkeeping stuff? Everyone's good? Okay. So let's get started with today's lecture. So today's lecture is actually a review of neuroscience. And so what I do is I usually put this, I do put this lecture in between getting into the motor control studies, because motor control studies are wanting to understand how we, or the sort of factors in relation to us controlling our body for actions or motor skills, correct? So we first need to remember how physiologically we control our body, and then how different factors, remember, was the environment, people, or the supporting surface and the skill itself. Like, remember like the different factors that affect someone's ability to perform a motor skill, how those different variables affect us controlling the body specifically. So today's lecture is actually a review of neuroscience, just super generally, and so it's not too long. I can go in as much depth as you would like, or we can sort of skim through, just as a show of hands, or whoever wants to talk, or I don't know if there's a function to like do anything, but who took neuroscience last semester with me? Me. Well, hit it. Go. I saw one didn't. Okay. So then let's just like, okay, so we have 2 no's and about 1 billion me's. Perfect. Okay. 3 no's. So for Elena, there was Marcus, I think, and someone else said not me. Oh, Marco's taking it this semester. Okay. Well, this will give you a good review. Well, you know what? And for those people that haven't taken neuroscience yet, so Dr. Murphy's using my slides, so it'll be transferable. Don't worry. But I guess this would be a good sort of, I'll keep it really, like I said, intro, just so that you have, for those students that haven't taken neuroscience yet, they'll have like just a general understanding of how we physiologically move the body. Keep that in mind, because we need to understand how to control that machinery when understanding motor control. Does that make sense as sort of like a comment or a objective with this lecture specifically? Okay. So for Elena, Marco, and anyone else that hasn't and Violetta, just if there's anything that you don't understand, follow along. Okay. Yeah. I mean, I don't think any of the so the other thing is, based on the midterm, the majority of the questions are not from this lecture, because again, this was review, so that'll help you guys as well. So this is just a good review. I think there's like 6 or 8 questions out of like 50 on the midterm from this lecture in comparison to the others. So this one is not heavily tested on, and I'll highlight the most important points. Okay, Marco says it's fairly simple, and again, this isn't like a crazy in-depth overview. This is just generally hitting all the different points. So let's get into the review of the neuromotor basis of motor control. So imagine how we physiologically, our brain is able to control voluntary movement. Oh, okay. And so what we're going to do is break this lecture down into 4 different components. Understanding the neuromotor basis of movement, talk about the neuron, which is the functional unit of the neurological system. We're going to talk about the peripheral nervous system and afferent versus efferent flow, like so sensory versus motor. And then we're going to finish off with talking about the central nervous system, which is sort of like the spinal cord and in the cerebrum or the brain, and how those are sort of initiating voluntary movement. So remember that the neuromotor system is our foundation for the control of movement. That just means that our connection of nerves to the muscle is how we voluntarily control our bodies. We control our movement from the top down. The human nervous system is divided up into 2 different sections, or 2 main sections. We have the central nervous system, which is everything encased in bone. So that would be everything in the skull and the vertebral column. So that would be our brain, our brainstem, or cerebellum, and as well as our spinal cord, everything encased. So that's called the central nervous system. The peripheral nervous system is all of the neurological structures that are outside of the vertebral column. So that would include all of the spinal nerves that are exiting at each level of the vertebral column, for like C1, both sides, C2, and all the way down, and how those reconnect to create all of our peripheral nerves, things like the sciatic nerve and the median nerve, the radial nerve, and our autonomic ganglia. Those are all in the peripheral nervous system because they're outside of the vertebral column or outside of bone. Okay? So when understanding the neuromotor system, it's the part of the neurological system that is specific to controlling voluntary movement or coordinated voluntary movement. So the review of this lecture is going to be looking at specifically those components. So let's first start off with the neuron. The neuron is the functional unit of the neurological system. And so it's this specialized cell sort of like extrapolated out that creates our ability to send one signal from one area of the body to another. That is the purpose of the neurological system. And the neuron is a highly specialized cell, and there are specific components that sort of differentiate it from other animal cells. So things that are differentiating of the neuron that make it a neuron are that it has 4 specific components. The first one is the soma, which is also defined as the cell body. Now, specifically with the with neurons, the soma or the cell body is what is sort of synthesizing all of the neurotransmitters or the proteins that are used at the synapse of the neuron to send a signal to another neuron or another cell. So in terms of the cell body of the neuron, it's highly specialized in creating neurotransmitters or like sort of developing them and packaging them and then sending them out to the presynaptic terminals. The dendrites are the input to the cell. They're like branch-like extensions from the cell, and that acts as the input to the neuron. And so if you imagine the neuron as a big wire or like an HDMI cable, the input would be the dendrites. The wire component is what's called the axon. The axon is highly specialized for sending an electrical signal through voltage gauge channels. And then the end of, or like the distal end of the neuron is the presynaptic terminal. The synapse is the connection of one neuron to another cell, typically another neuron or a muscle cell, for example. And so the presynaptic terminals, the before synapse terminals, are the end of the axon. And that's where we have a release of neurotransmitters. So let me show you a picture. Oopsie. Yo. Bam. Okay, so this is a drawing. This is kind of really what a neuron looks like. But we see the dendrites, the input of the cell. That's where there's connections to either receptors or other neurons that are going to send signals and accumulate them. This is the cell body. This is where neurotransmitters are going to be packaged and created. And then we have the axon. An action potential is sent down the axon to the presynaptic terminals or the axon terminals. Does that make sense, everyone? If the focus or the purpose of the neurological system is to send a signal from one area to another, the functional unit that allows us to do that or allows the body to do that or the neurological system to do that is the neuron. The neuron acts as like an electrical wire. Everyone good? So the synapse, like I said, is the connection from one neuron to another neuron or a neuron to another cell. So here we have like 3 neurons being connected together. The presynaptic neuron, the before synapse sends information, so it delivers information. The postsynaptic, so the cell after the synapse, receives information. So sometimes that screws people up. Now, question for all of you, specifically those of you that took Intro to Movement Neuroscience. What is responsible for sending a signal from one neuron to another or from one neuron to a cell across a synapse? Well, so the action potential goes down the axon, hits the end, and then the action potential doesn't cross the synapse. So then what, like, yeah, exactly. So it's neurotransmitters. So so that fact sort of delays the signal, right? If it was just one wire, it would be a lot quicker. You have to go from a signal down to the presynaptic terminals. The neurotransmitters have to float across the synapse, interact with the postsynaptic neuron, accumulate local potentials, generate enough potential for an action potential, and then that signal continues. So you're not wrong, Rayuta. It's just that the action potential reaches the axon terminals and then causes the release of neurotransmitters. We good? No, we're not good. We're good. Boom. Okay. And so through the 4 structures that we've talked about, neurons are able to receive information, integrate it, transmit that information, and transfer information to another cell. So it's like being a little computer, and it's not that it's a microchip. It's because the dendrites, right, the input of the neuron, they receive many different sort of local potentials, right? So when the neurotransmitters get released, or if there's receptors, we have each binding causes a little bit of a potential. And those local potentials all sort of add up to create our threshold potential, which will initiate an action potential. So we have the summation of local potentials at the dendrites, right? Whether it be from receptors or neurotransmitters. We have a buildup of local potentials, and when there's enough of them, then we get the threshold potential to cause an action potential. And the action potential is an all or none response. And so this process of summation of local potentials is the way that, because the neurons are getting constant information, constant, like it's always sort of integrating excitatory and inhibitory potentials, right? Some neurons are saying go, some other neurons are saying inhibit, right? So it's integrating information by this like basic principle of summation, either temporally or spatially, that allow for a buildup of a charge. And if there's enough of a positive or an excitatory input, the threshold potential is reached at the neuron level, and then that causes an action potential. So it's through this way that we say that the neuron is able to integrate information. It's not like a computer chip, but it is summating all of the different sort of information that's being input into the dendrites. So with spatial or temporal summation, so that's called general summation, we can have local potentials that are going to add up temporally. So the same local potential location is being activated multiple times, or if we have multiple local potentials occurring different sites of the dendrites, those will all sort of add up. So here we have temporally, we can see that the charge is building up, versus if we add spatially, we get a much larger response than would be if just we had one local potential. So it's the accumulation of all of these different inputs, whether or not some of them are inhibitory, some of them are excitatory, some of them want to cause an action potential, some of them want to inhibit it. As long as we look at how much it's all accumulating, like it's all summating or like adding up, then we can understand it, whether or not there's going to be enough of a stimulus to generate an action potential. Does that make sense, everyone? Specifically those that have not taken Intro to Movement Neuroscience yet? Violetto, Marco, someone else? Who was it? Spatial is many small. Yeah, so spatial and temporal are the same little small ones. Spatially means it's like a bunch of little ones happening at different locations at the same time. Temporally means you have little ones occurring in the same spot in quick succession. Oh, you did the lecture this one. That's awesome. So did I do it like you get it? We did a better job. Don't tell me. Tell me. You guys okay? Okay. Cool. Oops, sorry. So now what we're going to do is now we're going to move on to so that was the neuron. That's the functional unit of the neurological system. Now let's talk about the peripheral nervous system. The peripheral nervous system, like remind you guys, is all of the nervous system structures that are outside of bone. So that's outside of the brain and outside of the vertebral column. So if we looked at every level of the vertebra, at each level there is a spinal nerve exiting. So that would be the beginning of the peripheral nervous system. Now, if you looked at zoomed in as how these are sort of entering the spinal cord, axons or the nerves can flow in one of two directions. Afferent axons carry information from the periphery up to the spinal cord. So afferent, that's like, so I don't know if you guys know this acronym. I know afferent because it arrives at the brain. So that gives me the direction. Whereas the other way that the information can go is efferent. So that would be information that's going from the central nervous system out to the periphery. And that would be our motor system. So sensory information is afferent axons or afferent information that goes from the periphery to send to the spinal cord and up, versus efferent information is that information that's being sent out from the spinal cord out to the periphery. Cool? Okay. So let's first talk about our afferent axons or our sensory neurons. Now, typically, they look, so the most common sensory neuron is a pseudo-unipolar cell. So if you look at this diagram, it looks a little bit different than the hypothetical neuron that we did in the previous couple of diagrams, or the diagram from several slides ago. Now, a pseudo-unipolar neuron looks like this. Can you see how the cell body's out to the side? Now, the reason why this is because the fact that the cell body's out to the side, it means that we have the information that can be transmitted from the dendrites out to the axon terminals. It doesn't slow down when it reaches the cell body because it can be all transmitted. Like if the cell body was right in the middle, you would have an action potential that would come down, slow down to get past the cell body, and then continue on again. Having the cell body out to the side, it just speeds up flow. Now, in terms of motor control, in terms of the way that we sort of coordinate our body, think of it as sensory information is always the first. If you want to decide how to move, you need to be aware of your surroundings or what's going on. So sensory information is always primary. So our body has put precedent in understanding the sensory information. So that's going to give us information from our environment, from all of our sensory receptors, our eyes, all the touch, temperature, vibration, proprioception. All of that information is going to be sent up afferently from the receptors, from the periphery, up to the spinal cord, and then up to the brain. Now, the cell bodies of our sensory neurons, or the cell bodies of our pseudo-unipolar cells, are located in the dorsal root ganglion of our spinal nerve. So I don't know if you guys remember this from neuroscience, but here we have a sort of drawing of the information from a spinal nerve coming into the spinal cord. So remember, peripheral nerves are mixed. They have afferent and efferent flow, those neurons that are sending information out from the periphery up to the spinal cord, and then those that send information from the spinal cord out to the muscles. If you look at the spinal cord, all of the sensory information enters through the back end, through the dorsal root. And so this like sort of like enlargement is called the dorsal root ganglion. It represents all of the cell bodies of our pseudo-unipolar cells, of our sensory neurons, which means that the sensory neurons themselves can be like 1.5 meters long if they're at the legs, for example. All of the motor information, so all of the efferent information, exits the spinal cord because it's going out to the body. It exits through the ventral root. So the spinal nerve is mixed, but all of the sensory information enters into the back end of the spinal cord. All of the motor information and exits from the ventral side or the anterior side of the spinal cord. So it's organized. So when we're talking about the efferent flow, that would represent motor information. So what would be information that we're sending out to the body? Activation of our muscles, right? We're sending out a signal that is ultimately going to get to the muscles and cause them to contract. If we're talking about controlling our body, right? The whole point of this course is to understand how we control motor skills. So we are aware of information through our sensory system. Our body's able to process it. Wait, what's going on in the environment? What do we need to do? How do we need to move? What do we got to do? Right? Calculates it and then sends out a signal. That signal is sent out through efferent axons, which are coming out the ventral or anterior side of the spinal cord, and then go out to the muscles. So there's two types of motor neurons. There's alpha motor neurons, which make up the motor unit. The motor unit is how we activate our muscles. So it represents a alpha motor neuron, the lower motor neuron that exits from the spinal cord, and then the specific muscle fibers that it innervates. So if you think about, like, let's say hypothetically my biceps, let's say it has 1,000 muscle fibers. It's not just one neuron. It's not one motor unit. The way that we sort of determine whether or not we will activate a muscle by a little bit or a lot or a stepwise fashion is by recruiting more and more motor units. So for each given muscle, there is a certain number of motor units. So let's say in the biceps, there's like 1,000. If I wanted to flex it by 100%, I would activate 1,000 motor units. If I wanted to activate it by 10%, I would activate 100 motor units. So a motor unit represents one alpha motor neuron and then the specific muscle fibers of a muscle that it innervates. And remember that everything is like the activation of our lower motor neurons is an action potential. So it's an all or none response. So once you activate an alpha motor neuron, that action potential goes down and will cause a contraction of the muscle fibers that it's specific to. When we activate our muscles, it's not like all or none. Like it's not 100% biceps or 0% biceps. We can do it like in a stepwise fashion. And that's because all of our muscles has a number of motor units to innervate it. And so you can selectively, the body figures out selectively how many of the motor units to activate depending on the intensity of the contraction that you need. Now, one thing that I wanted to mention is that proprioception is very, very important, specifically for understanding motor control. Proprioception is provided by muscle spindles. Muscle spindles are units that register length and rate of length chains of our muscles located within the muscle fibers. Now, if you think of like a tape measure, if you have a tape, like a ropey tape measure, like a sewing machine, I don't know, people that sew, that have like a tape measure that is floppy. If you want to measure something, you make it tight and then you measure, like the fabric or whatever you're doing, right? So it's the same thing with muscle spindles. So muscle spindles have to be under tension to register how long the muscle is. So once you flex your muscle, because the muscle spindles are attached to the muscle fibers, they become floppy. So for every alpha motor neuron, there's a gamma motor neuron, so that runs parallel. So if we flex a muscle, we at the same time flex the muscle spindles at the same time. That is simply just to make sure that the measuring spindles, the measuring receptors are staying tight so that they continue to register length despite how long or how short a muscle is. And that process is called alpha gamma coactivation. So, is that confusing to everyone, or is that okay? Specifically, those that haven't learned this before. You guys are good? So Marco, and then for Violetta and the other one, sorry. Do you guys know what the term proprioception means? Oh, okay. Okay, well then whatever. Perfect. Let's go. Let's talk about something else then. Right. So our awareness in space is highly important, right? Like when we're doing motor skills, as someone becomes good at something, you don't have to, yes, exactly, spatial awareness. So if I close my eyes, I know how, like if I'm flexing my arm, if I'm extending my arm, I can feel that. And that's because the muscle spindles are telling my brain how long the muscle is. And then it calculates that, okay, well, that means that your body position has changed. You're aware of your body position without having to see it. And that's through proprioception. Proprioception is actually occurring because of these muscle spindles that send information. There's sensory neurons that send the muscle spindles send information to the brain. Now, the thing with muscle spindles, because they're so important, is that they need to remain tight. The only way to keep them tight is when you flex a muscle, you also flex the muscle spindles. Motor neurons, the alpha motor neurons flexes the muscle fibers. The gamma motor neurons flex the muscle spindles. Boom. So if the lower motor neurons are those neurons that exit the spinal cord, so remember, do they exit out the back end or the front end? Motor is coming from what end of the spinal cord? The front. Yeah. So we have all of our lower motor neurons that exit from the front end of the spinal cord. Now, the lower motor neurons are all arranged in what's called lower motor neuron pools. And so this is a labeled diagram. It's a picture, obviously, that's made up. But the way to visualize it is that the collection of cell bodies for the lower motor neurons to a given muscle, they're all sort of organized in what's called lower motor neuron pools. So if you look at the diagram on the slide, let's say this is an arm. So this would one would represent like biceps, pec, deltoid. Give me another muscle. Pectineus, serratus anterior. They're all sort of organized together. So the key with the neurological system is because there's so much processing that has to occur. The theme is heavily organization, organization or being heavily organized. So that's lower motor neuron pools. Now, if you think of the motor system and the processing that has to occur, even given what we've learned so far in this lecture, what you know about the neurological system and intramotor neuroscience, and what you're going to learn for those of you in the next slides, it's so much processing. It's crazy. And so the way that the body has sort of helped or figured out how to be able to control stuff is that a lot of functioning of the neurological system is involuntary. So it's controlled through interneurons. So the ratio, which is, I think, interesting for all of you guys to remember or understand, is that for every one sensory neuron, you have 10 motor neurons, and you have 200,000 interneurons. So interneurons are those neurons that relay information between and like sort of relay or divide up information, relay information back. They're involved in reflexes. We have so much relaying of information between neurons, and it's through the use of these interneurons that we're able to decrease the amount of processing that's required from the brain. A lot of it is done in sort of reflexively. And so here we have a diagram of potentially interneurons are those between the sensory and motor neurons. So if we have one sensory neuron, 10 of these, we would have 200,000 of the interneurons that are just relaying information back and forth. That sort of like help with reflexes, they help with coordination, they help with neuromodulation, all of the different processes that can be done sort of automatically that take away from the processing required from our brain. Am I going too fast? Are you guys okay? Again, this is just a summary. You're not tested heavily on this. I will give a breakdown of what questions or how many questions from each lecture for the midterm. You guys are good? Violetta? Mark was good? Good. Okay, so that was the peripheral nervous system. Now let's talk about the central nervous system. So the central nervous system is everything that is encased in bone. So what would that include? That would include our cerebrum, our brainstem, our cerebellum, and our spinal cord. Here we have the first component, which is our cerebrum. So that's what is like stereotypically called like our brain. This is what it looks like. So this is the sort of like landmark anatomy or topography of the cerebrum. Now, when learning neuroanatomy, and I think I discussed this obviously in Intro to Movement Neuroscience, when learning neuroanatomy, it's different than learning origins and surgeons of muscles. The way that you do it is that the folds, like the brain is like gushy, like it's gooey, like it moves around like jello, right? So it doesn't look exactly the same in everybody because it could be just jiggling around. What is the same between individuals is the location of these folds. So sulci are the indentations, when gyri are the folds. So we have a sulci, gyri, right? So the landmarking or in that landmarking for neuroanatomy is understanding all of the different folds. And the folds tell us, sort of divide up the brain into different components. Here we have the cerebellum, and then within it would be the brainstem. It's the folds that divide our brain up into different lobes. What we see is that the brain itself is divided up, or the cerebrum itself is divided up in two hemispheres, the left and the right, with a longitudinal fissure. The entire surface of the cerebrum is covered in gray matter. Gray matter represents the cell bodies of the neurons in the cerebrum. And then deep to that is the myelinated axons of those cell bodies. And so the white matter in the brain represents the axons or the tracks of those neurons. So let's look at a cross section. Here we can see a cross section of the cerebrum. Here we see the gray matter on the outside, and then the white matter is deep. So the way to visualize it is that if each of these represents, so if the gray matter represents cell bodies, the white matter represents all of the wires that are going down or up, like all the wires going up and down from the gray matter. So these are cell bodies. This is all the wires. Now I had mentioned sulci and gyri as how you do, or how you utilize, you landmark the cerebrum. Looking at the sulci and gyri allows us to divide up the cerebrum into different lobes. And we divide the cerebrum up into different lobes because we divide up the cerebrum based on sort of general function. So here we can see the four main lobes of the cerebrum. We see the frontal lobe, the parietal lobe, the occipital lobe, and the temporal lobe. And I've listed what they typically do. In terms of this course and Intro to Movement Neuroscience, it was really important to understand the central sulcus, which is the division between the frontal and parietal lobe, because the parietal lobe is sensory right here. So this is the sensory information where it gets processed, and then this is motor information where it gets sent out. So the central sulcus is a big landmark to divide up motor from sensory, which is important to this course, but also heavily important to Intro to Movement Neuroscience. Occipital lobe is important for visual processing, and then the temporal lobe is really important for memory, abstract thought, and judgment. Everyone okay so far? Because now what we're going to do is we're going to look at the cerebrum and look at the sort of part of the frontal lobe that is specific to the control of voluntary movement. So when we control, so we're talking about motor control in this course, obviously, we want to understand how we control movement from a neurological perspective. The way we control movement is from the top down. So we coordinate what muscles, or we motor plan in the motor planning areas. And so those would be the primary motor cortex, the premotor cortex and the supplementary motor cortex. This is the area that, based on a thought from the anterior part of the frontal lobe, we make a decision on what we want to do. This is the area, or these are the motor planning areas that help sort of process specifically what would be involved in the movement that we want to complete. So what muscles do we have to contract, what core muscles we have to contract, and what sort of things that we need to be sort of preparing in the background. That's completed in real time by the motor planning areas. And these are the ones that we're going to be talking about. The first one is the primary motor cortex. So it is located directly in front or anterior to the central sulcus. And the primary motor cortex is involved with the initiation and coordination of voluntary movements for fine motor skills. So the primary motor cortex is controlling our muscles for fine and dexterous control. Now, do you guys remember the term motor homunculus? This is a graphical representation of the part of the primary motor cortex, the part of the body that part of the primary motor cortex is associated with. So you guys might remember this question from intro movement neuroscience, but why do we have such a big graphical representation for the hand and the face inside of the primary motor cortex? Yeah, specific, that's true. That's right, Marco. And it's for fine control. Because in terms of fine control, we need most fine control of our hands and face. So if we have more neurons going to those areas, we have more control. Think of like a marionette. If you had like 1,000 strings versus 4 strings, you could do more fine control with more connections. And so the reason why the motor homunculus has a greater representation for the hand and face is because we need more control there. We need fine control for our hands that sort of describes us as humans, that ability, but also we have fine control of our face for nonverbal communication, which is heavily important to communicate with each other, or throughout with each other, sorry. Directly anterior to the primary motor cortex is the premotor area. Now, the premotor area is right here, and it's involved in the organization of movements before they are initiated. So what it does is we decide what action we're going to do. The premotor cortex makes sure that based on what we want to do, we activate our core muscles. So remember last day when we were talking about EMG studies, and it was like the skill was the guy that had to do this, and he just had to raise his arm, and they looked at which muscles were activated first, and it looked like core muscles were activated first. Now that's coordinated through the premotor cortex. If we're going to move, we don't want to fall over. So what our body does is it contracts the core based on what we're going to be doing, so it's stable, and then we do fine control. So that's achieved through the premotor area. Does that make sense, everyone? And then the other thing that the premotor area is responsible for that's really, really important is, remember we talked about like one thing that defines us in terms of learning motor skills? What's the best way to show someone how to do something? Or sorry, what's the best way to teach someone, give it away. What's the best way to teach someone how to perform a specific motor skill? Visual. Because we watch and learn. Kids do it. Like, demo it, right? You demo it. So what is it in a demonstration that we learn? The part of the brain that is sort of learning or watching or sort of configuring our system based on a demonstration is the premotor area. We'll get more into that in a different lecture, but that's the part of the area or the part of the brain that does it. Cool. Now, the last motor planning area is the supplementary motor area. And so this area of the brain is involved in the control of sequential movements. So, the best example that I can sort of come up with. So the first thing it does is it coordinates bimanual skills. So skills that involve different movements with the hands, right? So that way you can do both at once. It's also preparing for things that are going in advance. So if you were to take so this is the example that I always give. If you take a pianist, so a person that plays piano, and they are playing the piano, if they see, like if a high note is coming up, I don't know if I'm saying this right for any pianists that are around, but if a high note is coming up, the pianist will subconsciously move their hand and change the position of how they're using the keys to accommodate for the fact that they're going to have a high note coming up. And they do that subconsciously. The area of the brain that is coordinating what's coming up, like coordinating with your movements in relation to what's going to be like required sequentially, that's the supplementary motor area. So these are the three areas. These are the motor planning areas that coordinate how to activate the muscles and in what and like by how much for whatever motor skill we decide we wanted to do. Is that okay, everyone? Everyone's good? Awesome. Okay. So that is the motor planning areas. Now we also have regulatory areas. So the two areas that are important in terms of our control of movement are the basal ganglia as well as the cerebellum. Now the basal ganglia is deep to, like, so if we know we talked about the gray matter, deep inside of into the lobes is our what's called basal ganglia, which is a collection of different nuclei that in combination work to regulate motor control by inhibiting unwanted movements. They're like regulatory areas. So what it does is it looks at sort of the movement that you want to perform. It looks at the output from the motor planning areas and then decreases or increases the activation of the muscles to ensure that the movement that you're going to perform is smooth. And the reason why we know that is because we look at disorders where you have a problem with the basal ganglia and look at the patients. And like Parkinson's disease is the big one. They have uncoordinated, so they have really jerky movements. Now, we learned from internal movement neuroscience that the basal ganglia itself has different loops. We had concentrated on the motor loop because the idea was understanding the control of movement. But the basal ganglia is also sort of involved in movement initiation, as well as processing spatial memory and selecting desired behaviors. It prevents undesired behaviors and it shifts attention. So that's not super important for this course. The big thing with the basal ganglia is that it helps regulate unwanted movements by regulating the activation of our muscles. So what it does is it just looks at what we wanted to do, or like the signal that what it does is the signal that's coming out from the motor planning areas, it has to go through the basal ganglia. Can I go up a couple slides? Remember this slide? See right here, everyone? So okay, so let's say this is the primary motor cortex, and these are all the wires that are going out to the spinal cord. Does that make sense? See how they all have to go through both sides here? Everyone see that? This is the basal ganglia. So the basal ganglia is wrapping around all of the neurons that are going out. And so the basal ganglia will look at the signal that we're sending down and say, oh, no, adjust this so that way it'll be smooth movement. I'm obviously making it sound a lot more simple than it is, but that's generally what it does. I had mentioned Parkinson's disease. Parkinson's disease was a big topic in internal movement neuroscience, and we know about what the basal ganglia does in part because Parkinson's disease is something that has been studied for ages. We talked about in the course some of the symptomology that we get with Parkinson's disease. So we get slow movement, which is called bradykinesia. We get akinesia, or a reduced amount of movement. We get rigidity of muscles of the muscles, and we get tremors. Now, those are all symptoms, and we talked about in the course of what specifically or how those symptomologies occur based on the connections or the way that the basal ganglia motor loop works. And I don't know if you guys wanted to watch this, but just for interest's sake, I included the videos, the clinical videos from that lecture into this course. Do you want to watch them, the review, or do you want to keep going? Keep going, okay. Okay. What up, Drew? Okay. Now let's talk about the cerebellum, because the cerebellum is also a regulatory center. Now, if the basal ganglia is a way to inhibit unwanted movements, the cerebellum is, the way to understand it is, it's heavily important for this course because this is the part of the area that we learn coordination. It is heavily responsible for coordinating movement as well as postural control. Now, we had talked about all of the sensory information. The way that this works, and I don't want to get too much into it, but we have different cerebellar tracts. The cerebellum isn't conscious, but we have information that comes from our proprioceptors as well as our interneurons that go back up to the cerebellum. So the different tracts are sending information from all of our proprioceptors and looking at every time we flex a muscle, there's an interneuron that sends a signal back up to the cerebellum that we're contracting that muscle. Okay? And so what it's doing, or the cerebellum, is it compares our body position versus what muscles we activated. Okay? And then how do we learn motor skills? Through repetition. So every time we try to do something, if we were closer to the skill, like let's say we're learning how to pitch a baseball, let's say we're more accurate that one time, the cerebellum knows that this way that you contracted the muscles led to a better positioning of the body, which led to the goal. So it'll say that this one's better. And so it's the cerebellum with repetition that is memorizing what activation or what coordination or what order to activate the muscles led to a closer achievement of our motor skill goal. So motor pattern learning is in the cerebellum based on that. Okay, and then the bottom part of the cerebellum is responsible for coordinating equilibrium and balance. So generally speaking, the way to understand it is that the cerebellum in relation to motor control is involved in the learning, the timing, the rhythm, and the synchronization of movements. So in relation to this course, it's the learning and executing of stored motor patterns. So if someone has a problem with the cerebellum, they are uncoordinated. If someone has a problem with the basal ganglia, they have force problems with the muscles. Okay? Now, the last part of the brain that we haven't talked about is the brainstem. Now, the brainstem is the most evolutionarily oldest structure. So we see that a lot of the brainstem is related to like more like homeostasis type functions. So we have our medulla, which is a regulatory center for most internal physiologic processes. So heart rate, breathing rate, body temperature, like if not, you need to puke, like that kind of stuff. It also contains the reticular formation, which is sort of, it's integrating, it's like everything coming in and out of the brain has to go through the reticular formation. So when we have neuromodulators, like, when you have epinephrine and you get everything's all jazzed up and ready for fight or flight, that neuromodulator is sent to the reticular formation that's going to downregulate, for example, digestion, and upregulate like heart rate and things for fight or flight. The pons is just the connection of the cerebellum to the cerebrum, or to the brainstem up the cerebrum. And that's the brainstem. And then the thalamus, let me go back. Does anyone know what the thalamus is? Let me show you. I don't know if you guys know this, but see this right here? Right there. That is the thalamus. The thalamus is just like a relay center. So if you think of like a train station, the thalamus, everything going in and out of the brain has to first go through the thalamus. So the thalamus is like just a relay center. It sort of coordinates the signals going in and out of the brain. Oh, shit. Boom. Okay, so that was the cerebrum. Hopefully I didn't go too fast. Obviously, like, I'm trying to do a review, but was there anything that I touched on that maybe was too fast, or anything that you guys didn't understand specifically? I was gonna say, what you got? Okay, what is the internal feedback and high fidelity tract scan? So those are the spinal cerebellar tracts. So the internal feedback are those tracts that every time you activate a muscle, the feedback tract is an interneuron that sends it to the tract that sends that information up to the cerebellum. So your cerebellum knows what muscles you activated from the internal feedback tract. The high fidelity tracts, like high def, is those tracts that are from the proprioceptors up to the cerebellum. So the cerebellum compares the internal feedback to the high fidelity tracts. What muscles versus how your body's positioned. So they're specific to the cerebellum. Okay. Now we're going to go just into the rest of the last part of the central nervous system, the spinal cord. So the spinal cord is obviously more than just a sort of conduit from the brain out to the periphery. So much coordination occurs in the spinal cord through reflexes, through all of these interneurons. And it's important because it allows us to have less processing required in the brain. Having all of that information or ability in the spinal cord is what allows us to move the way that we do. Or else it would be too much processing and it would never work. If you take a cross-section of the spinal cord, this is what it would look like. So we've touched on this in the beginning part of the review lecture, and just looking at all of the sensory information coming from the spinal nerve comes in through the dorsal root, whereas all of the motor information coming out of the spinal cord comes out through the ventral root. Here we have the dorsal root ganglion. So this would be our spinal nerve, dorsal root ganglion, dorsal root, ventral root. Now, we had talked about white matter and gray matter in the cerebrum. It's actually the opposite in the spinal cord, where we have the white matter, which represents tracts or axons on the outside, and then we have gray matter, which represents cell bodies or unmyelinated axons in the middle, or in like deep. And that's all I need you to know about that. The way that, so when we talked about the lower motor neuron pools, how everything's sort of like all the lower motor neurons are grouped together into groups representing one muscle. Those, if you look at it from the top down, those muscles are going to be sort of grouped together based on the function of the muscle. So we notice that flexor muscles are more medial, proximal muscles are more medial, distal muscles are more lateral, extensor muscles are more anterior, and flexor muscles are more posterior in the anterior part of the gray matter. So just another way to highlight the fact that everything's sort of really, really organized. Okay. Now, the last thing that I wanted to touch on was sort of those tracks. And so if we go back up a slide, all of this white matter represents myelinated axons. And so those axons are grouped together based on all the axons for a particular function into what's called tracks or groupings. And so that was a huge part of internal movement neuroscience, was trying to understand how everything was grouped together, because it helps us understand how the body is coordinated or how we control movement by understanding how we have our sensory tracks, or that information that's grouped together and where it's located. And then we have our motor tracks, what they do and where it's located. By knowing where things are located, we can understand clinically what spinal cord injuries would look like, or why we have certain deficiencies with this injury versus not this injury. So let's first talk about the sensory tracts. Now, we have two main sensory tracts that are responsible for conscious awareness of our senses. We had our dorsal column, which was responsible for sending information from fine touch, proprioception, and vibration. And then we had our spinal thalamic tract that was responsible for crude touch, pain, and temperature. Now, Drew had talked about our spinal cerebellar tracts, so the internal feedback and high fidelity tracts. So these ones lead to the primary sensory area. It's our primary sensory area behind the central sulcus. That's where we're processing conscious sensation. So up to that point, it's just an action potential. It's our brain that processes that electricity as, okay, that means you have pain in your thumb. You have cold in your foot. Before that, it's just action potential. So these are ending up in our sensory system, or our primary somatosensory area. the spinocerebellar tracts are not going to the cerebrum, they're going to the cerebellum. And then this is how, like, when we finished off intro movement neuroscience, we understood the different sensory tracts. So here we have the spinocerebellar tracts. Here we have the dorsal column. And then here we had the spinal thalamic tract. So this is all sensory. Blue is sensory. And then we divided up our motor system into the motor tracts, which is what we're going to finish off the lecture with. Oops. Now, our motor tracts were divided up into the medial and lateral upper motor neuron tracts. So we had the upper motor neuron tracts coming up from the brain down to the spinal cord level where they exit. And then you had the lower motor neurons that would be the one or the neuron exiting from the spinal cord out to the muscle. So the upper motor neurons are the upper motor neuron tracts, and they were divided up into the tracts that we saw on the previous slide. The medial upper motor neuron tracts were those tracts that were responsible for posture and control. And so we learned them in the course, not very specific to this course, but they were the reticulospinal tract, the medial corticospinal tract, and then the medial and lateral vestibulospinal tract. So the medial upper motor neuron tracts were for core and posture. Just really generally, what sort of part of the brain was responsible, what motor planning area was responsible for core stabilization? Premotor, yep. So that would be an example of the part of the, like what tracks the premotor area would send out signals to core and posture muscles. Whereas the lateral upper motor neuron tracts were responsible for fine control. So that would be coming more from the primary motor cortex, right? The lateral motor neuron, upper motor neuron tracts are right here. Again, there's going to be no diagrams like this on the exam. This is just heavily for review purposes. So we're just all on the same page. Now, we had talked briefly about the whole idea of a motor unit. Do you guys remember that? Do you guys remember that? Like, does anyone not know what's meant by the term motor unit, where we have multiple motor units for any given muscle? Is everyone okay? Oh, did you hear that? Everyone good? Okay. Violetta? Okay. Last one, Drew. My guy, we're good. The one thing we haven't talked about in terms of motor units is that, have you guys heard of like slow twitch versus fast twitch? There's different like types of muscle fibers. All the fibers for a given motor unit are of the same type. So with training is that you're training motor units to become more slow twitch versus fast twitch. That would be the only thing. Because remember, it's an all or none response. So muscles aren't contracted by 0 or 100. They're by a stepwise fashion. And so the way that you can increase the amount of force in a given muscle when you're contracting is by recruiting more and more of the motor units for that given muscle. Now, the recruitment strategies, then when we decide to activate a muscle by more of an amount, is through what's called Henneman's size principle, which means or which dictates that we activate the motor units on the basis of increasing size. Smaller motor units are more endurance, whereas bigger motor units are for power. So you want to activate the endurance ones first in a stepwise fashion. The other way, so that's called increasing the motor units to increase the force of a muscle. The other way to do it that happens as well is increasing the frequency of motor unit discharge. And so this is really important for small muscles that only have a given number of motor units. Think of like the dorsal interossei or like the muscles that are responsible for your eyes. Like it's very, very, very small. So they only have a small number of muscle fibers and therefore a small number of motor units. You can increase the force once you've recruited all of the motor units. You can just take those motor units and fire them faster, and that will cause a little bit more force. So the way that we increase the force of a given muscle is by recruiting more motor units and increasing the frequency of motor unit discharge. And so the key point is that now that we've sort of reviewed all of sort of the like physiologic or neurophysiologic basis of movement, how like these are how the wires are connected, the part of the course what that course didn't touch on, the intro movement neuroscience, is where this course and where motor control research sort of started from, is the different, like, how is the brain coordinating all of this now? What's happening to control all of this machinery that we just discussed? And so the question is, how do we go from intent to action? And so I talked about this example last day by looking at MEG machine. Remember the magnetoencephalography? They were able to look at sort of, here's a hypothetical example of this study where they had someone in a machine either tapping their finger on beat to a metronome versus off beat to a metronome, and how that changed the motor pattern, or sorry, that changed the activation of the muscles in the motor planning areas. So when understanding motor control, it's not simply just how the wires and everything sort of coordinated, it's how we control the machinery, right? If you want to win a race, it's not just how fast the car is, you got to talk about the driver. Just made that one up. Does that make sense? And so what we want to talk about in terms of research is they wanted to look at the variables that are responsible of how, or what's required when we want to go from intent, so processing information, the intent of what we want to do, how do we use that, or what variables are involved in coordinating that before we send it out to like the neuromotor system. And so what we're going to learn in the course is so much of it is about processing. So we need to be aware of our environment. And as we get better at something, we're able to pick out what in the environment is most important, and we're able to process it quickly. And so this was sort of the idea of when looking at the motor system, this is like sort of the flow chart. What we want to look at is how we're controlling this. Now this is the end of the review lecture. Now, this is interesting. This was taken from like 2010, so a long time ago. Let me show you guys. Have you guys ever heard of brain interfaces? Anybody? Okay. So let me show you an older example. And it's not going to work. A bucket. Okay. Okay, let's watch it. It's only 2 minutes long. We have time.
A brain-machine interface is a system that allows disabled people suffering from physical disabilities, to communicate with the external world and also to control devices. What we are doing is measuring the spontaneous activity of the brain, electrical activity of the brain, and trying to detect patterns that are associated to different intentions. For example, if I imagine just imagining moving my right hand, this will generate a specific pattern of activity in my brain. Now, if we develop the software that can do the real-time analysis of what is going on in my brain, I can detect that particular pattern and transform that pattern into some external action. The practical possibilities that brain-machine interfaces offer to disabled people can be grouped in 2 categories: communication and controlling physical devices and virtual devices. One example is this wheelchair. In the case of a subject trying to control the wheelchair, what we do is to fuse the mental commands that are coming from the brain-machine interface with the intelligence of the wheelchair itself. This is called technically shared control. So for doing so, the wheelchair is equipped with 2 cameras that are perceiving the environment, so that every mental command is interpreted with respect to the distribution of obstacles.
To control the wheelchair, I use right hand and left hand motor imagery. This means that I'm thinking to move my right hand to turn right, or my left hand to turn left. And this feels very natural because it's a very simple task that everybody can do. At the same time, it's very quick. So after more or less in a second, I can deliver a command, then turning the wheelchair right or left.
What we want is to bring this technology out of the lab. And this has 2 components. First is testing with real patients, so as to demonstrate that this is a feasible technology that they can benefit from. And the second aspect of that is to guarantee that they can use the technology over long periods of time.
So that was like an older example of brain-machine interfaces. I mean, you guys are like, what sort of other things have you guys learned about that you guys want to talk about? I'm sure Elon Musk has one. But the idea with sort of understanding how the neurological system works in terms of motor control is that we can understand the sort of activation patterns that are required or sort of associated with different movements. So technically, we could use that information to control a machine. And so that's where sort of the research is coming from, either with prosthetics or like there was things with like brain machine interfaces with fighter pilots and like the technology there, right? Any questions, guys? Boom. Okay. So that was the review lecture. I'm gonna so the due date. I might have to put an announcement or change it on Canvas, but the due date for that first lab will be February 12th. And then I will. Oh, corona. Is there a link between depression and Parkinson's? There a little bit. It's more about so with Parkinson's, it is dopamine. Whereas sometimes depression is in relation to like serotonin and melatonin. But there's also other plays like, so if you are like, think about if you have like any disease or any sort of neurological disorder or any sort of, there's an anxiety or there's depression with that. So there's a link between depression and having a sort of like specifically a disorder that affects your activities of daily living. When someone is limited in taking care of themselves or their sort of independence, it leads to greater depression, or there's a link to greater depression, which would sort of make sense. Sure. So the reticular formation is just a network of, so remember how we had interneurons in the spinal cord that sort of help kind of relay information? The reticular formation is like all of the interneurons in the brainstem that are coordinating, sort of like, think of like when we have pain, we now are going to increase like stress hormones, or we're going to increase activation of flight or fight response. All of that is through the connection of interneurons called the reticular formation in the brainstem. Cool. That's good. Awesome. Okay. See you guys next week. Don't get the Rona. And I will keep you updated if there's any changes with the labs. Okay. Boom. Bye, guys.